Hey, Mr. B here. I'm going to be using a PowerPoint that was developed by Biorad. This is a picture of it, and I modified it significantly, um, but they had some good stuff and some stuff I didn't like, so I changed it. Um, this is going to be our end result. We're going to be doing another gel electrophoresis. We won't do this little fingerprint. That's just some fancy artwork, but this is what we will be doing. You'll notice, remember we had like a ladder. It was something that was like a standard. And then we're going to have some different samples that we're going to be testing. One will be from the crime scene, and then 1, 2, 3, 4 suspects. And so what we'll be doing is DNA profiling. Well, what's that? And that's using basically, in the end, ultimately, electrophoresis to tell one human being from another. And so what they say here, the unique genotype of each sample is called a DNA profile. So everybody, you, me, every person at school, around the world, they all have their own unique DNA profile. The only people that would have the exact same DNA profile are identical twins. But they also have some, I think there's something they can do there, I forget, but with the exception of identical twins, everyone has their own unique DNA profile. And so on the crime scene, as we've talked about, blood, and you could have, different human fluids, we'll put it that way. Here's some different things you could find. I can't imagine why someone would want to pee at a crime scene, but I guess, again, this was made by Biorad. And one thing this I put in here, just want to do a little quick review of these are chromosomes. Remember those guys? And you get one from mom and one from dad. So, they kind of pair them up and they always show them as X's, but really, this half came from mom, one half came from dad. And remember that each cell has all that information in it. And then also just remember that each chromosome is a big old strand of DNA. Alright, so that's what this visual is showing is here. Again, they're showing it kind of where it's going through cell division. And we're not worried about that at this point, but if you were to take that chromosome and ultimately you could unwind it, you'd end up with DNA. And what's important is that I'm start for the left-hand side. 99.9% of everybody has the identical DNA. So you think about it, it's like, wow, only a tenth of a percent of our DNA is different. So that genotype, we only differ by 0.1%. Pretty amazing. And so in genetic profiling, what they'll do is they're going to look in the genome, and they're looking for something that's unique from individual to individual, and then also anonymous. Now what that means is that that'd be something that doesn't control a trait like red hair or a function like making a protein for whatever we need, like collagen. We all need skin. Okay, so we're looking for these, you might call it junk DNA, but it does have some value to it. Okay, now in that chunk of this anonymous DNA, we're going to look for an area that's called an STR, which stands for short tandem repeat. Okay, so what we're going to do is we're going to look for, in that junk DNA, so to speak, we're going to be looking for STRs. Okay, so one STR is found on chromosome 11. And this is just, I just made this up, but let's just say that the STR region is here and there. Okay, so both sides of that chromosome will have that STR region. There was one here and one there. Okay, keep that in mind. And remember, one came from mom, one came from dad. And so the one STR that we'll be looking at is called TH01. What that means you don't have to worry about, but the concept of what this is all about. To get the concept, I need to get you a definition first. And the first definition is locus. And locus is a location on the gene. And what I mean by that is, so the locus would be like in here, okay? What I'm just indicating with the arrow. Okay, and that you've had that in bio, but I imagine it's been a while. You probably forgot what that was. You probably recognize it, but like, what's that? Okay, so well, now you've seen it. All right, So, okay, remember we're on chromosome 11. So this THO1 locus is something, as we mentioned before, it's anonymous, so it doesn't control traits or cellular functions. What it has in it is these repeats of TCAT, that being, thymine, cytosine, adenine, thymine. And so this would be the chunks of this THO1 locus. And what happens is it has repeats of this TCAT, 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 TCAT. Now what happens is this makeup, they all start with CCC, they all end with AAA, but what happens in the middle, how many repeats, that varies from individual to individual. And so this example had 6 TCATs, as I'll call it, and there's more than 20 known THO1 alleles. And alleles, you might be saying, well, what the heck is that? Well, I'll show you what an allele is in the next slide. Remember, you as a person inherited allele from each parent. So now you might be starting to get a concept of what allele is. If not, here we go. Okay, allele is one member of a pair of genes. Okay, so this yellow things will make sense in a second. Mrs. Smith, let's say we just randomly pick some lady, okay? Her locus for our 2 chromosomes are going to be here and there. She's got mom's chromosomes are here, dad's chromosomes are there. The allele is both of these guys together, okay? So that's the allele. It's a member of a pair of genes. So remember, you've got the mom chromosome, the dad chromosome. But what you'll notice is, okay, of course, mom's traits were different than dad's. So that is pretty interesting because what happens is, you're going to start getting some different combinations here that start making it easier and easier to identify an individual, in other words, do DNA profiling. So what I mean by that is, okay, so if we had a sample from Mrs. Smith and we ultimately had enough DNA to do a gel electrophoresis, so let's say this is our gel. This would be our allele. Okay, this would be the wells would be up here. I didn't draw them in there because I just didn't want to. I'm just a little bit lazy, sorry. Um, But let's say that this is our ladder here, that standard ladder like we did when we did gel electrophoresis. And then this is Mrs. Smith's sample. Remember, this would be the negative, and this would be the positive. And so remember, we had the negative electrode and DNA being negative, so it's going to get repelled. And it's going to take longer for long DNA to travel through this, through this, through the gel, and the shorter chunk of DNA would travel faster. So mom's chromosome, being much shorter, would travel faster. Dad's chromosomes, being longer, more base pairs, would take longer. Okay? And then we could use this information in our fancy software to figure out the lengths of base pairs. Right? So what we do is we take a sample from a crime scene, and from a crime scene, you're not going to get enough DNA to actually run the gel electrophoresis. So that's where this thing PCR comes into play, okay? We'll have some target DNA, that's DNA from the crime scene, maybe a little drip of blood, not enough to run gel electrophoresis. So what we need to do is we have a template and we want to make multiple copies, and so we're going to do that. We're going to make billions of copies using PCR. Okay, and so PCR, what it stands for is polymerase chain reaction, and that makes lots of copies. And what we want to do is we want to make a copy of a specific piece. Um, in the case of our lab, we want to make a copy of the THO1. All right, that's our goal, and so allows you to make multiple copies of that specific piece of DNA. And this is kind of an interesting analogy. PCR is like looking for a needle in a haystack. So say you found that needle, but then we want to mass produce that. So that's what it allows. It allows you to make a haystack out of the needle. Kind of a weird analogy. I think PCR is like looking for the needle in a haystack and saying, hey, I got a needle. I want to make a billion needles. And so you do that. um, So right now this whole PCR thing is kind of nebulous. Well, let's go to a really awesome visual. I got excited when I found this. This is the coolest. All right. So this is from the University of Utah, and I think they did a great job on this. So we're going to start out and go ahead and get that site at home. It's pretty cool. Okay, so they're talking about the human genome. We're 3.2 billion base pairs. That stuff you knew about. And in the cell nucleus, okay, that of course is where our DNA is. And what they're showing is in a PCR, our goal is to mass produce a little chunk, that one locus, and we're looking for that STR, in this case THO1. Okay, so how does that work? Well, let's go on to our, I get a little fancy here. All right, so let's say that on the crime scene you found a hair. What you'd need is the root there, okay? The word is slipping my mind, but you know that little white part that gets connected to your hair. My brain, this time change is killing me. Um, so anyway, so we take this little sample here, and what we would do is we would have a little bit of DNA. There's a little more work involved in getting the DNA from the root of hair, but let's just say you got that DNA from there. And we're going to be using your cheek cell next week. This week we're going to be utilizing samples that are provided to us from a crime scene. Um, and so what we do is we're going to have, this is going to kind of simulate our lab for tomorrow. We'll have a bucket with ice and another bucket with little tubes. It doesn't quite show it here, but really the tubes are really dinky that go into this machine over here, which is called the PCR. And I think I've shown you that. Okay, so we're going to get started. So first thing, I'm going to be taking my micropipette, and I want to take, this is, in this file here is going to be my DNA, okay? I want to get my DNA, and I want to put my DNA into a PCR tube. Boop. Okay, so now I need to add this thing called a primer. A primer is a really special chemical, which the visual is going to show you that later. So for now, we're just going to say, and by the way, primers are engineered. So I'm just going to go get this thing called a primer with visual later. We'll show you what that is. I'm going to put the primer in here as well. And then keeping in mind, I'm going to put in another primer. What the primers are basically, remember, if you remember really quickly, if you think back or if you have your notes, that visual of the STR started with CCC, and then at the very end it ended with TCA. Well, so the other primer is going to be designed for the other on the TCA end. Okay, so I'm going to go get the second primer here, suck that up and put that in there. So I put two primers in, and now if I want to make multiple copies of DNA, well, I'm going to have all the nucleotides, the A's, C's, T's, and G's. So I'm going to need to get some of that in there, so let's shuck that up and put that in the tube. Alright, now the one last thing that we need to make multiple copies of DNA is polymerase. Now polymerase is, needed to make copies of DNA. If we used our polymerase in this, it wouldn't work. The polymerase that we're going to be using is called Taq polymerase, TAQ. And the TAQ comes from the species, I think it's an algae, if I remember right, from Yellowstone Park. Yellowstone Park, you have those big geysers. They're very hot. And what they found is that there's actually algae there, and they grow there, and if they can grow, that means that they can replicate, and that means that they have polymerase that can handle very hot temperatures. And why we need the hot temperatures will be important when I show you this visual coming up. But just keep in mind, okay, so this polymerase came from an algae, I believe, in Yellowstone Park. And so it's called Taq polymerase. And I'm going to grab that sucker right there. Whoop, and we're going to pop her in. Bam, as Emerald would say. And by the way, if you notice, it's pretty cool. They zip off the tips. Every time we're gonna be going through a lot of tips tomorrow. Okay, you don't want to mix up DNA from suspect A to suspect B, obviously. So just keep that in mind when we're running this lab. Okay, so now the PCR has all our chemicals in there. It has the DNA from the crime scene. It has two primers, one for the CCC end, one for the TCA end. It has nucleotides and not most important, but last but not least, the polymerase, the Taq polymerase. All right, so let's go on, and these will be little things here. All right, and this is our thermal cycler, and we're gonna put it in to our thermal cycler, and then we're gonna click on start, and this thing is gonna run for about 3 hours. Now what we're gonna do is show you, well, what happens inside, because we won't be able to see it. So here is our target sequence. Okay, so our goal, remember, we're going to have that whole chunk of DNA from the crime scene. What we care about is this STR. What we really care about is the THO1 sequence. All right, that's what we care about. Now keep in mind, you're going to have one from mom, one from dad. Okay, so we're just focusing on, let's just say this is mom. All right. So here's mom's DNA, and it has that chunk that we're very interested in, that allele. Okay, remember allele because mom has it as well as dad, but they'll be different. And so let's focus in on the moms, and what we're going to do in the PCR is, first of all, it's called a thermal cycler, and that's because it cycles a temperature. So it starts out and heats up to 95 degrees C, which is about 200 degrees Fahrenheit. Now, if you ever had a temperature of 203 degrees Fahrenheit, you would be having a really bad day. Well, what is important about that temperature is that what it does to the strand is it unwinds and unzips. Now, keep in mind, in that solution, we had the primers. Okay, so what we'll do is it's going to cycle. It's going to cool down to 50 degrees C. And at that temperature, the primers, let's say this is the primer for the CCC end, it's going to pop on. And let's say this is the other primer for the TCA end of this, the THO1. So they'll be complementary base paired. They'd have to be, otherwise they won't be attracted there. And so cools it down to 50 degrees C. Not enough to have the two strands come back together, which is pretty cool. And so then next, we're going to heat it up a little bit. It goes up to 72 degrees C, which our polymerase wouldn't be able to do this. Our polymerase would break apart. That's why we have to use this Taq polymerase, TAQ. And so the Taq polymerase is attracted to this end of our primer. And then what it's going to do is do what it does best, and it's going to copy. Woo! And it cranks. Notice it cranks all the way to the other end of that chromosome. Bam! All the way to the end of that, well, I should say DNA. So it made a huge chunk of DNA. And, if you're thinking, you're like, well, I thought we really care about this. And you're right. We do really care about that. But just hold on a minute. So now what we're going to do is we're going to heat it up again. Ooh, baby. Unwind, unzip. The primers are still in that solution. We're going to cool them down. The primers, they will attach. Polymerase will do its thing, okay? Now notice, we had from the original template, the original chunk of DNA, we made an exact copy of itself. But notice this time, what you ended up with when you did the copy the second time around, I'll go back one, watch what happens right here and right here. It made the target sequence. We really care about that length right there and right there. Alright, that's pretty interesting. So now we'll repeat, heat up, unwind, unzip, boom, do the whole thing. Okay, and so now we're starting to make the desired fragment size. Okay, let's do it again. Heat up, cool down, etc., boom, boom, boom. And now all of a sudden you'll notice, okay, yeah, we've got the original length of DNA here and here, kind of like an intermediate lengths, but we're starting to get more of the target length. Well, let's just repeat the process. Let's cycle again, thermal cycler. Bam, bam, bam. Wow. All of a sudden, hey, what's going on? We're starting to make way more copies of our target sequence than this other stuff, which is nice, but really not what we care about. And we repeat that, repeat, 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 and wow! We have made, in the end, over 1 billion of the fragments that we really want. Yeah, you've made a lot of, the DNA of lengths that you really don't care about, but not that many relative to 1 billion fragments of that target DNA, in our case, the TH01 sequence. And so this billions is what we're going to be running in our gel electrophoresis. And so what we'll be doing in the lab, we're going to have an allele ladder for us that's just going to run in gel electrophoresis, which would be down below. That's going to be, let's see, if we're doing Wednesday, it'd be Friday. Okay, so we'll run our gels on Friday. What we're going to have is a vial that has crime scene DNA, and then we're going to have a vial from 4 different suspects. So we've arrested some people and collected DNA from them, and so these would have been where the DNA is already prepared. In the future lab, where you take a cheek cell, you're going to be making your own DNA. So we'll go through that process in the future. For today, it's already provided for us. And so what we're going to do is we're going to take what's called master mix. The master mix. Aha, funny. Yeah, master mix is basically has the nucleotides, it has some buffers, it has the polymerase, it has the primers in there. Both you have needed 2 primers, so it's all going to be in there. So it saves you a bunch of steps of micropipetting. And so we're going to put these in these little, wait till you see them. These things are really small. These little, the PCR tubes are dinky little things. They're like cute, but they're really small. And then we're going to put them in our PCR, and we're going to let that run for 3 hours. And then once we have that going, we're going to do something pretty cool. It's a little self-directed, but it's cool. It's doing a little analysis of a pathogen outbreak. So we'll be working that while we do labs here, because we'll have like 40 minutes for lab, but then 40 minutes we'll be working on that pathogen outbreak. So that's what we got coming up for ourselves. A couple terms here really quick. So that heating process, they call it denaturing. The cooling process is called annealing. That's where we're putting the primers on that template strand. And then warming, and that's where you're going to activate the Taq. There it is. You can see it, Taq polymerase, and it repeats 35 times. Okay, so not that it's super critical. Remember that it repeats 35 cycles, but at that point you've made billions of copies of, in our case, the TH01, and then we'll be ready for gel electrophoresis, which we'll do on Friday. All right, so that's it. That's all I got to share with you. And we'll go from there. Thank you very much.